Hi there, Dr. McCoy with Piedmont Reproductive. I'm here today in our embryology lab in our Greenville office, and this is where everything starts. From having the egg retrieval in our operating room, the eggs get then processed and fertilized by our embryologists on these microscope systems. Once they are fertilized, they go into our incubators, and we have state-of-the-art incubators. Each incubator here has separate small sections that holds one patient each, so they're all kept separately. And they are cultured up until the 5th day. Culturing them until the 5th day really allows them to sort themselves out into which ones are more likely to be able to make a pregnancy. Looking at the embryo stages, they start to divide on the 1st and 2nd day. By the 3rd day, they have about 8 cells. By the 5th day, which is really as long as we can keep them outside the uterus, they have formed a blastocyst, which has a fluid-filled sac, and it's starting to separate into the cells that make the embryo and the cells that make the placenta. At this point, they are frozen. And the way they are frozen, they are placed on a little device such as this. This is called a cryo-lock device. And this is a small little device that it's a little hard to see here, but it's basically a little plastic wand. And one embryo is placed on each one. It's then plunged into a tank of liquid nitrogen and immediately frozen by a process called vitrification. Once those are frozen, they are then stored long-term over here in our lab in these liquid nitrogen tanks. They are frozen at minus 197 degrees Celsius, which is about minus 320 degrees Fahrenheit. And once they are frozen, we really think that they can last really indefinitely. So why do we freeze embryos? In the old days, we used to do fresh transfers, but that was really because our freezing technology was not as good. Nowadays, the freezing technology is extremely good. Survival rate after freezing is above 95% and really should be quite higher with good embryos. The main reason we freeze and don't really do fresh transfers anymore is that, one, it's the safest thing for the patient. It really has mostly allowed us to eliminate what was termed ovarian hyperstimulation syndrome. And two, it also allows us to freeze them, which lets us create a better environment. When we are stimulating the ovaries, we have a lot of hormones, and that doesn't give the best environment for the uterus. So we can stop there, let all those hormones wash out, and then come back and get the uterus ready in as best shape as possible. Doing genetic testing allows us to weed out the embryos that aren't any good genetically before we freeze them. And that way we know that what we have frozen is the embryos that really have a chance. Many couples are concerned about making embryos that they may never use. And it is a small portion of our patients that actually have more embryos than they will ever use. There are steps that we can do to mitigate that. You can not fertilize as many embryos or as many eggs initially to limit the number of embryos and the concern about that. But really, it's only a small percentage of embryos that can actually make a pregnancy anyway. And so genetic testing has really helped let us determine which ones are potentially viable. So that's the process in the freezing steps in the lab. We'll see you soon.